Hello and welcome to Connect Ovar Coffee, where we host candid conversations, make meaningful connections, and talk about all things ovarian and gynecology cancers. I am Runsi, the founder of Overcome, and today we are joined by a very special guest, Dr. Debra Richardson. Dr. Richardson is the Division Chief and Professor of Gynecologic Oncology at the Stevenson Cancer Center at the University of Oklahoma. Regarded as one of the leading experts in the early phase clinical trials, it is our absolute honor to have Dr. Richardson with us today. And she is in her full glory of the Overcome scarf and looking beautiful and gorgeous. Thank you, Dr. Richardson, for gracing us with your presence. And we look forward to learning a lot from you in the next 45 minutes to an hour. And as we go along, as you are watching, if you have any questions, please put them in the comment sections below, and we'll try to get it addressed post the discussion. We'll share the questions with Dr. Richardson, and as and when we receive responses, we will post it back online. So with that, a huge welcome to you, Dr. Richardson, to this episode of Connect Over Coffee. An honor to have you with us.
Thanks so much, Runsi. Really happy to be here today.
Thank you. So I have ton of questions because you are absolute treasure when it comes to the early phase clinical trials. So before we get there, I just wanted to, just to level set our foundation. I know that you spend a lot of your time developing and leading phase 1 clinical trials. So can you tell us a little more about the phase 1 trials? Many of us, like even clinicians still associate phase 1 trials with a little bit of uncertainty and a lot of toxicity. And so, how do you reframe that value proposition when it comes to those trials today?
Yeah, that's a great question. I kind of break it down. So in general, there are kind of two parts to phase 1 trials. The first part is really the dose escalation. And so that's where we start at a very low dose and then, gradually work our way up. And each trial has its own design of how many levels will be investigated. And that's usually based on preclinical data and animal models before we put them into human beings. The second part of a phase 1 is a dose expansion. And that's where we're trying to get a little bit more idea of the efficacy of the drug and also the tolerability. Phase 1 trials have really changed a lot from even when I started my career to now. And I think we do it better. And I would say there are a lot of safeguards in phase 1 trials to really try and keep patients safe, because that's the primary objective is really safety. But the second thing is to understand how well do these drugs work. And the FDA started a project called Project Optimus, and much of that is being done in phase 1 now. And really that's saying, instead of going to what's the highest dose we can put safely into a person, really what's the optimal dose that we should be using, especially with these novel therapies. We don't necessarily need to go to the max dose. We need to go to the dose that balances both how effective the drug is and how tolerable the drug is. Some phase 1 trials are for all comers, and some are restricted to certain tumors or biomarker driven. Patients are really monitored very closely. There are extra safety checks, extra lab draws, especially early on. Cycle 1 and cycle 3 tend to be kind of the longest, most visits for a patient, and most lab draws, and sometimes very long days and those kind of things. And we keep very close check on our patients on phase 1 with really close communication between the nursing team and the patient. And I will say that a few of my patients, I started in phase 1 a little over 8 years ago, about 8.5 years ago. And a few of my patients from then are still alive. And I really believe that they are still alive because of the phase 1 trials that they participated in. And so I feel like phase 1 trials offer the possibility of getting an active drug years before these drugs become commercially available.
Thank you for mentioning that. It's very important that you raise that point, that especially with your patients who are doing well past 8.5 years of being in your trial, that is definitely hopeful. Thank you for sharing that. So when it comes to biomarkers or any molecular signatures that are proving most useful right now in selecting patients for early phase trials for gynecology cancers, what would you share with us in terms of that?
Yeah, this is a really busy area right now. So I would highlight, I think the ones that we understand the best are really the BRCA mutations, homologous recombination deficiency, especially when we're talking about ovary cancer, the folate receptor, HER2/neu, which could be for any gynecologic cancer, the mismatch repair status, sometimes for ovary cancer, probably more commonly in uterine cancer. There's a very specific mutation of the p53 gene, which is the most common mutation that we see in cancers as far as p53, but they have a very specific mutation that they're looking for called the Y220C. And we have a trial right now. It's actually in phase 2 called Rosetta Pop is the drug. And then there are also trials that are not selected by biomarker, or at least not in the beginning, but other biomarkers that we're kind of investigating now, especially for antibody drug conjugates, include cadherin 6 or CDH6, TROP2, Claudin 6 or CDLDN 6, and then B7H4. But there is a plethora. I couldn't even list all the different biomarkers that we're looking into at the moment.
So when it comes to the biomarkers, just going back to it for a second, as you said, there are a lot of biomarkers that are now and emerging. And then so how is in your experience, how is the testing doing in terms of, biomarker testing in patients? And I don't mean only in big academic centers such as yours, but, even in community setting hospitals. What is happening in terms of the biomarker testing? Do you see gaps? And if so, how do you envision us bridging that?
I think, the first step really is all patients with ovary cancer are recommended to undergo germline testing. So hopefully all patients with ovary cancer at the minimum are being offered germline testing. Yeah. And then there's tumor testing. And in general, especially for advanced ovarian cancer patients, really their tissues should be sent for what we call next generation sequencing, which has come a long way. There are many commercial products out there. People use different ones. For example, there's Caris, Foundation, Tempus, Myriad, etc. In addition, some institutions at academic centers have their own in-house testing. But in general, we need to assess those things. And some tests are actually by immunohistochemistry, so not necessarily the next generation sequencing. And some of the tests that you can order will do both. They'll do both immunohistochemistry because, for example, the folate receptor, that's an immunohistochemistry test. HER2/neu, also typically an immunohistochemistry test rather than a sequencing test. Others, like the p53 mutation, you can stain for it, whether it's present or not, but if you're looking for the specific mutation, that's something that requires molecular testing. So hopefully that's being universally applied. I am aware that it is not being done for everybody, but I would say definitely in the last few years, the huge majority of patients are having this done up front. So I think we have come a far way in making sure that our patients are being offered these tests and that they're being done.
And thank you for mentioning that, because if they are not done, then you have given us that information. They have, empowered us with that knowledge so that patients can now go in and ask their providers. Because keeping in mind that, we typically, our episodes go out to globally. It's not just here in the US. So it's important that patients everywhere that are watching know that these things need to be done. So thank you so much.
Welcome. I will say sorry to interrupt you, but internationally, I would say it is different. And I would just recommend that they then speak to their doctors, because what is going to be available will not be the same country to country. And oftentimes the United States gets the drugs first. So for example, mirvetuximab is not available in every country in the world at this point. It's definitely more and more countries are coming online with mirvetuximab, but even now, not every country has the ability to use that drug or to test for the folate receptor. So there is going to be some differences depending on where people are watching from.
Thank you. So moving on to the next question. So when it comes to staying on early phase trials, because you are an expert in this space, so when it comes to those kinds of trials, what are usually your key considerations and criteria when you recruit patients for these studies? And how is the integration of immunotherapy and targeted agents changing the structure and endpoints of these phase 1 trials?
Sure. So, each trial has a checklist, I like to call it, of inclusion and exclusion criteria. And many of them overlap, but there are differences. And so, whenever we're considering a patient for a trial, we have to go through that checklist. And sometimes a patient wants to go on a trial and the doctor wants to put them on the trial, but they don't meet all the eligibility criteria, unfortunately. So, I always do let people know that's a possibility. But in general, a patient will have to have had the standard care therapies expected to give benefit before they would be eligible for a phase 1 trial. The labs have to be within range, and that's going to be defined by the trial. But in general, we're looking for a good, what we call absolute neutrophil count, which are those white cells that help protect against infection, hemoglobin, which if it's low means that somebody has anemia, and the platelets have to be high enough. We're also typically looking at kidney function and sometimes liver function as well. In general, most patients are going to have what we call measurable disease, and that's based on CT scan. So for example, PET scans usually cannot be used to enroll a patient on a trial for GYN cancers, at least especially ovary and uterine. And so that means that usually a tumor has to be at least a centimeter, or a lymph node. We look at the short axis or the shortest dimension, and that has to be at least 1.5 centimeters. We also look at how many prior lines of therapy a patient has received before. We assess the performance status, and that's kind of how much the person is able to be up and about during the day. And in general, most trials are going to say it has to be either a performance status of 0, which means that they're able to do all their normal activities that they did before they were diagnosed with cancer, or a performance status of 1. Immunotherapy trials may use a modified RECIST known as iRECIST, and that's to take into account that sometimes immunotherapies can cause what we call pseudoprogression, where the tumor looks a little bit worse, but our imaging cannot distinguish between tumor and our immune cells. And when you're trying to traffic immune cells to a tumor, it could actually make it look fluffier before it gets smaller. So not every trial that uses immunotherapy uses iRECIST, but many of them do. And so we do keep that in mind as well. But a patient would have to be doing clinically well. So if a person was not doing as well clinically as they had, say, when they started, and we see that the disease looks worse, then in general, that is still considered progression, even with immunotherapy. And, we're right now trying to find the right dose, like I mentioned before, rather than the maximally tolerated dose in regards to endpoints.
That makes sense. Thank you. So, as I mentioned, you are at the forefront of so many exciting initiatives. So, tell us about the clinical trials you're leading or involved in, and any details that you would like to share with our overcomers for them to know in terms of even like considering signing up for these trials.
Yeah, I'm happy to talk about this. So for one thing, I am the national PI of a trial called GOG 3096, or Rejoice, and that is not just in the United States. It's also in Europe and some places in Asia. This is a phase 3 randomized control trial of a drug we call raludotatug deruxtecan. That's a mouthful. Or we can shorten it to call it RDXD.
Okay, much better.
Much better. Versus investigator choice chemotherapy, which is weekly Taxol, PLD, which is some people call that Doxil or topotecan. It's an antibody drug conjugate that targets cadherin 6 or CDH6. And in both our phase 1 and phase 2 trial, the response rate was about 50%.
That's
I think yeah, it's really great, actually. And it's pretty well tolerated. It can cause some decreased counts. And it also can cause something called pneumonitis, which is inflammation in the lungs. So it's something to be aware of and has to be monitored for. But I think this is a really exciting trial that's out there. And I would recommend that any patient with platinum-resistant, high-grade serous ovary cancer think about this. We do allow 1 to 4 prior therapies before. And then I'm also the national PI of a trial called GOG 3124. Right now, that's still in phase 1, and it's investigating a drug called IMGN 151, which is a novel folate receptor antibody drug conjugate with a microtubule toxin. It's made by the same company that made mirvetuximab, or some people might call that Elahere.
Okay, so you talked about the 2 trials that you talked about. They are both ADCs, right? But
that is correct.
But different types of ADC. So help us understand that with this explosion of ADCs in the market, right? We understand the folate receptor alpha as the benchmark for, whether or not the patients are going to benefit from the treatment or not. But with all of these ADCs coming into the market and there with the subtle differences in between them, is it appropriate to say that it's then going to help more patients regardless of their folate receptor status? Or, I mean, how would you integrate all of these ADCs into the system?
Yeah, that's a great question that we don't have a good answer for yet. What I would say is many of the antibody drug conjugates coming may not require biomarker selection, or they will be eligible for many more patients than, say, mirvetuximab. So, the FDA approved mirvetuximab to be used for platinum-resistant ovary cancer for patients whose tumors express folate receptor in a high fashion, so greater than or equal to 75%, 2 plus, 3 plus.
Yeah.
These newer generation appear to be more generalizable, so they can be used for more patients and at lower folate. So there's another drug out there called Rina-S, and that's a antibody drug conjugate that targets folate that has a different type of payload. So mirvetuximab uses a microtubule toxin. Rina-S uses a topo-1 payload, and that drug, at least so far, has not required a biomarker. So in other words, it doesn't require pre-selection. And we're seeing that quite a bit with many of the new antibody drug conjugates that are under investigation. So it kind of remains to be determined. I think there's going to be a lot of questions. Right now, like our antibody drug conjugates, we think about what's the target and also what's the payload. And we just don't know how many antibody drug conjugates a patient is going to be able to receive. And there's a lot still to learn about what the resistance to these drugs are. And so that might help us better understand, okay, actually, in this case, it wasn't a resistance to the payload, it was a resistance to the biomarker. And in other patients, it's going to be resistance to the biomarker. So use a different biomarker, but you could still use a similar payload. So there's a lot here still that we have to learn in regards to sequencing.
Sounds like it. And then, just curious question on this, staying on it for a second, that between the PARPs and the ADCs, then, like, when the PARPs were coming in and we had 3 or 4 different kinds of PARPs, and it was from the conversations I've had with the physicians, they were like, no matter, so there are subtle differences in their side effect, the mapping. However, it doesn't mean that if you start on one PARP and you go to another PARP to go to another PARP, you will, your reaction or response to it will probably be the same. I mean, regardless of the side effects. So I'm kind of bringing it back to the ADCs, that with so many ADCs coming into the picture now, and I think you said we don't know the answer yet, but for a patient to go from one ADC to the other, to the other, to the other, if that happens, we don't know how that patient is going to fare under those changing ADC scenarios. Is that correct?
That is correct. I will say that there's a couple. So, for example, with the folate receptor antibody drug conjugates, I'm aware of at least 5 that are under investigation, but there is limited data. So there's one made by Lilly. They do have some data in post mirvetuximab exposed patients being treated on their ADC. There's a little bit of data being generated on that for Rina-S, and I can't speak for all of the antibody drug conjugates. So we are trying to understand that. Right. But right now, in general, you will see that if you have had an antibody drug conjugate that has a topo-1 payload, you often won't be eligible for another antibody drug conjugate with a topo-1 payload on trial, with some exceptions. So I don't want to make a general statement, but that's often the case. Until we do some more work, I think, quite frankly, in both phase 1, and then also once these drugs become available, we're going to have to do some trials of trying to understand sequencing. Should we give this ADC first and this one second? Should we swap that? How do we choose? There's just a lot. It's an explosion, time. And then we're going to have to do a lot of the hard work to figure out how do we choose the best drug for each patient.
Exactly. That was my question, and you answered it candidly and beautifully that we don't have all the answers yet. However, with all these, this is hopeful that so many treatment options are coming in, but in what sequence and which ones to give and how to personalize, that still remains to be learned as we go. So, thank you. So, as a clinical trialist, we would love to have your help to understand this whole journey of the drug development from the lab to the clinic, right? So we understand that if the trial is positive, it goes to phase 2, phase 3, and then there's an FDA approval, and then finally it goes to the clinic. But oftentimes we get asked by our overcomers, especially if a clinical trial or a study is showing a lot of promise, that how how long does it typically take between these phases to the FDA approval to finally for the providers to receive the drug and actually prescribe it to their patients? If you could just walk us through that timeline.
Yeah, that's a great question. In general, I would say it's a minimum 6 to 7 years and can be longer. Some estimates are as long as 10 years, depending on the drug, the disease, et cetera, from our first phase 1 trial to FDA approval. And then, approvals are different throughout the world. So each country has their own approval process. In the United States, once a drug is FDA approved, it can usually be used fairly quickly thereafter. So for example, we just have had 2 recent approvals in ovarian cancer that's platinum resistant, both the B96 regimen, which is the weekly Taxol with or without bevacizumab with Pembro, and that you can pretty much use now because all those drugs were already around. There can be a slight delay of insurance companies catching up, but they're usually also fairly quick, and we can go through an appeals process if needed. And the most recent one is, of course, the relacorilant, the Rosella trial relacorilant with weekly nab-paclitaxel. And relacorilant is also now been, they've manufactured it and it can be used. There can be like a slight delay trying to get that into your institution, but I would say at this point today, people could start receiving those treatments. So in the US, that's pretty fast. My understanding, for example, in Europe is even after the EMA approves it, there's still just because it's approved, then there's a delay in their insurance, their healthcare system, making it available. So each country is going to have their own variation of how long that is from approval to actually being able to get it to a patient. In the US, it's fairly fast once we approve it.
And then to your point, first time you open a phase 1 trial to the point where we eventually get the FDA approval, it could be between 6 and 10 years, depending on. Okay.
That's true. Yes.
Okay. Thank you. So as we were talking about, the trajectory of ovarian cancer is probably the most hopeful that we have seen so far. So what are some of the novel treatments and approaches that are emerging that you think has the potential to alter the landscape of treatment and change the outcomes for?
I would say,
but yes.
We've been talking about antibody drug conjugates quite a bit. I think that is definitely something that is helping outcomes. And as we have more available, we'll continue to help outcomes for our overcomers. I would also point out bispecifics. That's something that we don't talk about a lot, but I am kind of interested in this. Bispecifics means it targets 2 different things, typically the tumor. So it has a protein target on the tumor and then also the immune cell, trying to traffic the immune system to the cancer to help your immune system recognize the cancer is foreign and kill it. And we've had a lot of issues trying to get immunotherapy to work very well in ovarian cancer. And I'm a little bit hopeful that this might be a way forward for that as well. There are also many biomarker driven, science driven phase 1 trials of combinations. Sometimes it's hard to give 2 drugs together because of overlapping toxicity. So trying to really get newer generation drugs that reduce those overlapping toxicities and drugs that can be safely combined. But there's a lot of thoughtful science going on to try and figure those things out. So I think there's quite a bit coming that really is exciting. But right now, I'd say the things farthest along by far are the antibody drug conjugates.
Okay. Thank you. So I was reading about ctDNA, so I'm going to bring you to that. So, how do we incorporate these tumor biopsies, the ctDNA, or other liquid biopsy platforms to understand the early response during the phase one trials? Help us understand the background behind all that.
Yeah, great question. We also don't know, but I will say in general, like some trials do mandate checking, for example, ctDNA, and they each do it at their own interval. So sometimes you might get the ctDNA checked when patient is like treating the first day, right? So we call that cycle one, day one, and then they will, collect it at various time points during the clinical trial, and really to kind of understand, does that help predict? For example, can we predict earlier who is going to be a responder to the drug, who is not? Because if we could do that, maybe we would say, okay, your tumor is not going to respond. We should stop this now and find a different treatment for you, because that could spare toxicity from a treatment that's not going to be effective. So that would be a great thing if we could do that. We're not there yet, but that is something that we're, curious about. That's how I would say, like, most phase one trials are really trying to understand at this point the ctDNA. Like, there's different ways to use ctDNA. One of the, commercially available, for example, ones is really looking for what we call minimal residual disease. So it's potentially more sensitive at detecting disease than our tumor markers and CT scans. The question is, can we make that actionable? Can we take that information and then change therapy for a patient earlier and help them live longer or improve cure rates? Right now, again, we don't have that information, but that is things that we're very interested in learning about. So sometimes ctDNA is used during maintenance. Sometimes it might be used at new diagnosis to follow treatment. But there is a lot to learn here.
And just for everyone listening, if you can just briefly explain what ctDNA may be.
Sure. So that stands for circulating tumor DNA. There are different ways to do it. Some are just an assay that does the same test on everybody's blood, looking for markers that are known to, be expressed on tumors. Others are more personalized, where they actually take the patient's tumor that has been resected in the past and sequence it, and then do a blood draw and look for any evidence of that person's tumor circulating in their blood. So there's different ways to look at ctDNA.
Okay, thank you. So, I have personally talked to patients who had to sign out of phase one clinical trials due to side effects, toxicity, and they just could not tolerate. And so, these are, and as you said, like 1 to 4 lines of therapy, typically, usually heavily pre-treated patients. So, and you talked about those escalation safety just at the onset. So, what would be your guidance in balancing that dose escalation safety with the urgency? Many patients feel that when standard options are exhausted and they enter into the clinical trial, phase one clinical trial, with the hope that this would be the answer that they've been seeking.
So I think that's a really important question. I think goals of care discussions are really important between a physician and the patient, because we all want everyone to live as long as possible with as much quality of life as possible. So I really do encourage everyone to, talk to their providers about side effects and what their goals are. And we often have options to hold the drug, dose reduce the drug, do symptom management. If we don't know about the side effects, we can't manage them very well. So really, like, open and honest communication is really important. And I think shared decision making here is very important. So I'll often have a very frank discussion with the patient, asking about what their goals are and what their side effects are and what's the most important thing to them. I would also say that I don't necessarily look at phase one trials as waiting till all standard options are exhausted. There's a difference between using the ones that are associated with improved overall survival, for example, like some of the regimens we spoke about earlier, versus, yes, we have options, but we know they're not very effective. And these phase one trials sometimes may offer more hope and better efficacy than what would be available off of a trial. And I also look at standard options are always there, whereas trials may not always be there. If we think about how far we've come in pediatric cancer outcomes, about 60% of kids with cancer go on clinical trial. Compared to adults, it's under 10%. And there are many reasons for that. But I do think that the way that we improve outcomes in cancer is by clinical trials.
Wow, I needed to pause on that for a second. You said that 60% of the kids are actually going to clinical trials versus adults, which is less than 10%. That is
Yes.
a big gap.
It's a big gap. And if we look at how that has improved both cure rate and how long, children live with cancer, it's tremendous. And we can only make that kind of progress if we have more adults participating on trial.
Thank you for sharing that statistics because sometimes, data can sync, it really sync in with all of us. So this was one of the data points that you shared that I'll definitely stay with it for a while. So, I was talking about AI, like thinking about AI because everybody else is thinking about AI or, doing AI, talking about AI. So how do you see this AI or the predictive modeling influence the dose selection, toxicity reduction, patient matching, most importantly in phase one trials? You talked about that a little bit over the next 5 years, or is it already being used? And where is this? What is the status of AI in phase one?
So that's a great question. I'm not an expert in AI, but I will say there are some programs out there already using AI to match patients to trials.
Okay.
And I do think that AI is going to have a profound impact on drug development and many aspects of the medicine. I do think that AI will probably help us figure out toxicity prediction. Also, can take complex information and then perhaps it'll say, well, yes, the tumor has this mutation and that might say that this drug would help, but then it has this mutation, which will say that the drug is actually not going to work. I think it might help us better pick treatments for patients in the future. Right now, that's limited, but I think we're going to see in the next 5 years an explosion of that.
That would be wonderful because, while we definitely do not want AI to take over the medical world, but at the same time, just doing this kind of based on big data, if they are able to tell you or guide you, and then we, like you were talking about, we spare a particular patient from the toxicity because it's obvious that this drug is not going to work for her. And so, I mean, I can see so much. This matching in particular would be so great, and then patients can go on to flourish in that particular phase one trial because they're all matched to it. So that would be wonderful. We look forward to that happening. So switching gears a little bit on talking about PARP, and so I understand there's this whole primary versus the acquired resistance in PARP inhibitors for platinum resistant disease. So if you can just give us a little overview of what is the primary versus the acquired resistance, and also just out of curiosity, with all of these ADCs now coming in, where is PARP being, where is PARP fitting into this puzzle?
So great question. I would say, platinum, like PARP inhibitor resistance basically means that the tumor just is not going to respond to a PARP inhibitor at all. And in general, those would be the homologous recombination proficient tumors. The issue is we don't actually have a test to determine if the tumor is homologous recombination proficient. What we have are tests to say, yes, this tumor has evidence of homologous recombination deficiency. But it's not 100% true that if a tumor doesn't test positive for that test, that it is homologous recombination proficient. But we in clinical, medicine kind of splits it at whether the test is positive or negative. In other words, if the patient's tumor is homologous recombination deficient positive upfront, then they are offered a PARP inhibitor maintenance. In regards to acquired resistance, that's somebody who was responding to a PARP inhibitor, and then the tumor eventually grows, say, on the PARP or potentially shortly thereafter stopping the PARP inhibitor. There are multiple ways that a tumor can become resistant to PARP inhibitors. The three main ways, and these are kind of complicated, but One is the drug target related resistance. For example, a tumor that was homologous recombination deficient is able to become homologous recombination proficient. So in other words, restoration of homologous recombination. And then there's also something called restoration of replication fork stability. These are kind of complicated concepts, but they are how the cells make repairs to DNA. I break it down in thinking like, okay, when you're texting somebody and you make a spelling mistake, you have autocorrect to fix that. So in our cells, when our DNA is making copies of itself, it has these ways of fixing the errors because it makes errors sometimes. And the most high, what we call fidelity or best way to do that is through homologous recombination. And when the tumor can't do that, it then has other ways, but they're not as good. And so PARP inhibitors cause that synthetic lethality in tumors that are homologous recombination deficient because it can't use the PARylation pathway either. And then there's so many errors that the cell has to go through cell death because it can't repair them.
Yeah.
But eventually resistance develops because it finds ways around that.
Okay, thank you. And
you asked, sorry,
yeah,
I
was going to.
about, I think PARP inhibitors still have a very important role. For frontline maintenance, if a person has a BRCA mutation, they 100% should get a PARP maintenance. That is likely improved with quite a much higher rate of cure than if somebody does not get PARP inhibitor maintenance up front. In addition, patients with homologous recombination deficient tumors in the frontline maintenance, that is still the gold standard. I don't know that we're ever going to replace that. It maybe we'll do combinations or things like that, but for now, at least, by far, that's the best we have for BRCA and for homologous recombination deficiency. Where I see antibody drug conjugates potentially fitting in would be for those patients whose tumors were not homologous recombination deficient. There are trials ongoing actually in frontline maintenance and also in platinum sensitive disease, either as a treatment instead of platinum or as maintenance after platinum. So that's where we're starting to see more of the antibody drug conjugate. So I don't think it's PARP versus antibody drug conjugate. I think they're different patient populations or different disease settings. And in fact, mirvetuximab is being studied now in neoadjuvant setting with carboplatin. So the question is, can we replace Taxol with mirvetuximab? But you would still have your same maintenance options.
Right.
So I don't think it's one or the other.
Yeah, and also thank you for breaking it down for us. I was just thinking, you answered that question, is there is no competition between the PARP and for the lack of a better word, PARP and the ADCs. They belong to separate spaces as of today, and then they could potentially also complement one another depending on how the treatments evolve. But there is no direct conflict that you have seen between PARP and ADCs. Okay.
That's correct.
Thank you. So I'm going to ask you about this ever elusive early detection of ovarian cancer. Not to say because you're in the early phase clinical trials, has nothing to do with its detection, but it's a question that for someone like you who's so invested in the well-being of the patient community, I feel like we are still being outpaced by the early detection is still not happening after these many years. So where is that window of opportunity that we are not still grabbing, you think?
Yeah, that's also a great question. And I wish I had a better answer, but I would say there's a few things. Part of the issue is the majority of what we call quote unquote ovarian cancer, high-grade serous ovarian cancer being by far the most common, actually starts in the fallopian tube and not the ovary. So a lot of our old work, we were misinformed because we were looking for screening on the ovary, but the tumor wasn't starting there. It was starting in the fallopian tube. And by the time the ovary was involved, that's already left the fallopian tube. It's left the primary organ. You're already talking about a more advanced stage cancer. But there are ongoing trials for ovarian cancer screening. This is something that people have not given up on. We just have to change how we're doing it. And so I'm not going to get into all the different trials, but there are actually ongoing trials today trying to do ovarian cancer screening. I would say to date, though, today, our best effort should go to prevention. And how can we do that? So birth control pills are a very effective way of reducing the risk of ovarian cancer. It reduces the risk about 10% per year if they're taken, and it lasts for at least 10 years after the birth control pills are stopped. So we do have some things we can do. There's also opportunistic salpingectomy. And what that means is when somebody's already undergoing surgery, that their fallopian tubes, if they're done childbearing, can be removed. Because again, most of what we call high-grade serous ovarian cancer starts in those fallopian tubes. And we are seeing, decrease in high-grade serous ovary cancer because of opportunistic salpingectomy. So if somebody is having their uterus removed, there is no need to leave the fallopian tubes there. If somebody's is done childbearing and wants a tubal ligation, for example, instead of tying the tubes, we can remove the tubes. Now, that can't be reversed, so you have to have a good conversation with your clinician that's going to be doing that surgery, but just to be aware. And actually, we're looking at, can we do this during other surgeries? For example, somebody's going to the operating room for hernia repair or their gallbladder removed. It has to be an elective surgery. It has to be when it's a safe time to do it, but there are probably many opportunities to remove women's fallopian tubes to reduce the risk of ovary cancer. And finally, my plug would be that we need to really do our best to identify previvors. In other words, people who have inherited mutations that increase their risk of ovarian cancer and other cancers. But right now we're talking about ovary cancer because there are prevention strategies for that. For example, risk reducing, salpingo-oophorectomy, or basically removing both of the ovaries and the fallopian tubes. So there are things we can do today to reduce people's risk.
And I feel like, when you talked about the familial history, and that's 20 to 25% of ovarian cancers that could be potentially genetic. Then, I have said this before in this program is, there's a whole subset of population too, like me, who don't carry the mutation, but we have a direct family history because my mom passed from ovarian cancer, even though I did not carry the gene, but that doesn't mean my risk is probably higher than the general population. And so there's a whole subset of us in between that need to be also managed. And so all the three techniques that you talked about are all actionable. And so we definitely need to keep those things in mind. And, because we still don't have although there are many clinical trials in progress, but we still don't have a reliable screening test. But these are some of the ways that we could potentially reduce the risk, especially for the at-risk population. So thank you so much for mentioning that. So we all love the concept of the precision medicine, right? But many patients, even from my experience, including my own mother, I have seen that we go through this whole cycle of therapies without seeing any clear benefit. And so what do you think is the disconnect between the molecular profiling and the actionable treatment, and what steps we need to take to actually overcome those? And in heavily pre-treated overcomers too, how do you distinguish between a drug that is truly ineffective versus that is being tested in a disease state that is too late in the whole journey?
So, yes, there are still many limitations. There's a few things. When you test a tumor, you may get many mutations. And so tumors have different types of mutations. There are both what we call driver mutations and passenger mutations. And what that means is a driver mutation is driving the tumor's biology. It's driving the tumor's function and ability to spread and survive. You have to target the driver mutations. Targeting a passenger mutation basically will not be effective and will give the drug toxicity with no, effect on the tumor. The problem is we don't always know. In some tumors, we know, okay, that is definitely a driver mutation. So the best example that just popped into my mind is, for example, the BRCA mutation in ovary cancer. That is a driver mutation, and we have targets for that. Another one would be in breast cancer. For patients who have breast cancer and it's HER2/neu positive, that is a driver mutation. But there are other mutations that can be drivers in some tumors and not in others. And so we don't always know, especially in ovary cancer, because ovary cancer can be very heterogeneous. In other words, it's really hard to put ovary cancer into categories because if you profile a bunch of different high-grade serous ovary cancer, they're going to be different. I do think we're getting better at treating ovarian cancer patients more personalized and not treating them all the same. We can start at the highest level of that. So, high-grade serous ovary cancer is very different than low-grade serous ovary cancer. So, for example, we've had the new approval for people who have low-grade serous ovary cancer who have KRAS mutations of the avutometinib defactinib. Okay, that's not a drug combination we would use in high-grade serous ovary cancer. There's clear cell ovary cancer. That's very different than high-grade serous ovary cancer. So I think mucinous also, we're getting better at saying, okay, your tumor is this histologic subtype. Let's not treat it all the same. And then also taking that molecular, data and trying to target that. But some of the molecular things that we find, we just don't have drugs to target that at all. Whether or not we will in the future, maybe. It kind of depends, I think, on how common they are and how druggable those mutations are. But sometimes, we don't get it right because, yes, the tumor has this mutation, but that's not what's driving its behavior. So targeting that is just not helpful.
Yeah. I mean, thank you for pointing that out, because you're absolutely right. There are so many subtypes of ovarian cancer, and even within the subtypes, there are unique differences between one person's cancer versus the other person. So to get to that level of granularity, it takes a lot. So, I mean, thank you for all the work that you all are doing to get us there, but I do definitely understand the complexity and the amount of research it takes to take us to that level where we are understanding and personalizing treatment for each and every patient based on her tumor type. So definitely not an easy feat. So there's also this increasing interest that I've been reading about in moving these promising agents that we talked about through the course of our discussion in earlier in the treatment lines. So what scientific or regulatory barriers, in your opinion, exist today that prevent this faster transition from phase 1 to frontline settings? And I would, ask you an ambitious question, is that as a phase 1 trialist, if you could redesign the entire phase 1 phase from scratch, what would you change to better align all this great scientific discovery with patient benefit?
Yeah, that's very ambitious, but I think I'll just start. I would say one thing I wish was different is that patients with ovary cancer who have non-measurable disease but detectable disease would be eligible for more trials. It's very rare for me to be able to get a patient that doesn't have measurable disease onto a trial, but many patients with ovary cancer never actually develop measurable disease. And so I feel like we're missing that part of the population where we're studying these drugs in the measurable patients, but are they the same as every patient that we're seeing, for example, with ovary cancer? So I'd really like to
So,
be able
Dr.
to.
Richardson, before you go into the detail, I want to understand the difference between the measurable and immeasurable disease. Please help us with that.
Sure. So as we had talked about a little bit earlier, measurable means that they have at least a centimeter tumor or that a lymph node is at least 1.5 centimeters in the short axis. Non-measurable but detectable. So a patient has a CA125 that's elevated, might have carcinomatosis, so has obvious disease on CT, but doesn't meet that criteria that has been set out by the research community on how we measure cancer. Might have ascites, might have pleural fusion. Those would probably be the most common things I'd be talking about for ovary cancer. But I have patients like that who have obvious disease, elevated CA125, carcinomatosis, ascites, and I can't put them on a clinical trial because they're not eligible because there's no tumor that meets the criteria for that measurable disease. And I really would like to be able to know, do these drugs work in those patients as well? Do we need to be doing something different for those patients? And so that to me is probably one of the bigger things that frustrates me when I'm trying to treat all the disease, all the patients I see with this disease. I want to make sure that we're including all patients to the best that we can. I would say, the bar is really high in especially like frontline ovarian cancer. I think a lot of us would really love to replace Taxol, at least in the front line. I would like to be able to use it for platinum resistant ovary cancer because it is a really effective drug. But we know that it causes hair loss, which is very distressing for people, and it causes neuropathy. And many times that neuropathy never goes away. And there are things that we've tried to do to reduce the neuropathy. Some are maybe a little bit more successful than others, but I still have patients who have neuropathy for their entire life after, Taxol. So could we replace that? But it's really effective, right? So I think the bar for replacing some of these things it either has to improve cure rates, or it has to be as effective as carbo and Taxol, for example, but reduce toxicity. And so that's a very high bar. So how do we do that? And we have to start thinking about that early on in the phase one, I think, time period to try and get as much information as possible to be able to do that. But we also don't want to compromise, like when we're already doing well, right? We don't want to hurt patients. So there is definitely some things that we need to learn in order to be able to move these drugs earlier, and see if that does change outcomes.
So, just a curious question on the measurable versus immeasurable disease. What percentage of patients that you see come to you with measurable disease versus not?
That's a good question. I think a lot of patients are sent with measurable disease because the referring physicians know. But I would say maybe 20% or so don't have measurable disease.
Okay, so we are missing out on those 20% of patients that we don't know the answer whether they would have done well on the phase one clinical trials or not. So that's a significant percentage of patients that we are missing out on. So
yes. Now some phase ones do allow them, okay, but some don't. I would say the majority don't, but occasionally they do.
Okay, that's good to know. Thank you. So we have we have discussed a lot of science over the past almost an hour, I think. So what have I not asked you that you would like to share, Dr. Richardson?
I think, we've covered a lot, like you said. I guess my plug would be that clinical trials offer hope and more treatment options, and they're how we make progress. So I really encourage everybody to think about participating in a clinical trial. And if you're not being offered clinical trials, ask your clinician. Yeah. Is it because the place that you're being treated doesn't offer them, or is it because there are things perhaps in your medical history or your tumor history that make you not eligible? But I do think that clinical trials really should be brought up to all of our patients.
I agree. And then I will also say that on our website, we have this whole clinical trials finder, so that, and it's only ovarian cancer. So any of our overcomers and our community members, if they want to go search for clinical trials in ovarian cancer, they can do so just directly going onto our website and finding what is happening and where. So, but yes, it is so important to have that discussion with your provider. And ask him or her of what the clinical trials that are available that could be beneficial to you. We definitely stress on that. So we are almost at the top of the hour. Before we close, though, this is one of my favorite questions that I ask all my episode guests, which is, what message of overcoming would you like to share with everyone that's listening today?
Well, I think this was a great time for us to meet because we just had 2 new FDA approvals. And really, we're seeing the pace of FDA approvals, I think, improve. They're faster and we're having more options for our patients recently for ovary cancer. So really exciting times with the 2 new approvals of both the relacorilant with nab-paclitaxel and the B96 of the weekly paclitaxel with or without Bev with Pembro. Plus, again, for those low-grade serous ovary cancer patients that are KRAS mutated, they've got the avutometinib, defactinib, which was last year, mirvetuximab in the last couple of years, and you're going to see more approvals coming in the next couple of years based on these phase 3 trials that are being done. So I think we're in really exciting times right now.
And thank you so much for that message of hope. And I would turn around and say that this is happening because of the tremendous work that you and your colleagues are doing behind the scenes to bring us from where we were. When my mom got diagnosed with ovarian cancer 15, 16 years ago, there was not much available. But now I'm so excited to see so many things that we can offer to our patients at this point in time. And it's just becoming that disease that it can. So the goal to me before we find a cure is to at least get to the point where it becomes a chronic disease where we can stay with it, we can enjoy the full quality of life and get treated just like other chronic conditions, but we are slowly but surely getting there because of the tremendous work that you all are doing. So thank you. And we are very grateful to you, Dr. Richardson, for all your time today. It was a great chat with you. We talked about so much science and so much hope. So very much grateful for your time. And thank you to all of you for joining us today for this episode of Connect Over Coffee. If you found this information useful, and I'm sure you did, please share this with anyone who may benefit from all the wisdom and the insights Dr. Richardson shared with us today. And we will be back with the next episode of Connect Over Coffee very soon. Until then, you keep inspiring and keep overcoming. Thank you.
Thank you so much.